great school, but it never got to UC Berkeley. You hear UC Berkeley and your jaw drops. Hunter College in New York, CUNY Hunter, isn't UC Berkeley. So it's not Harvard, it's not Yale, it's not any of those things, not even close. And this guy Wang, he's got a lab at UC Berkeley. I'm sorry, he's got a lab at CUNY. And no great drug has ever come from CUNY. In fact, really just publication-wise, not a lot of great science has come from CUNY either. We don't have too many Nobel Prize winners or anything like that. So it's just something where we struggle to attract that kind of talent, and a lot of it is funding related. And over time, maybe we can reverse that, but that's the sort of present and past state of CUNY for the last 30 to 40 years. So Wang's got this lab at CUNY, more or less, and again, my understanding of all this is somewhat fuzzy, but basically he's publishing, I read most of his work over his entire career, and he's publishing relatively basic neuroscience biochemistry work. He's not an actual chemist, keep this in mind. He doesn't know medicinal chemistry. He doesn't know how to design drugs or design medicines. I would venture to say my medicinal chemistry skills vastly outweigh his, for example, but that doesn't mean I'm a great medicinal chemist. And I think that I'm not even sure there's a chemist around that lab, which is, it's fine. It's not a drug lab. It's a neuroscience basic research lab. Okay. So eventually, the story that I understand, and again, if there's others that understand better, I'm all ears. Basically, what ends up happening here is there's an effort to sort of think about naltrexone, naloxone, and these other sort of opioid binders and look into them. And these are very well studied drugs. I don't know for sure, but I'm pretty sure if you go to the Rutgers PDB, you can find co-crystals of naltrexone, naloxone with each of the 3 opioid receptors. There's this mu, kappa, and delta. These are all well understood. There have been inordinate numbers of attempts to tease apart the efficacy and the side effect liability of each receptor. People have tried to make partial agonists to each specific subreceptor. If you just do kappa antagonist, what happens? If you just do a kappa agonist, what happens? Then do the same for mu, then do the same for delta. And the amount of medicinal chemistry that's been published in this space is just enormous, right? This is one of the most fundamental kind of like parts of pharmacology that exist. So somewhere along the line, this protein called filament A comes in. And it's not a protein that's particularly exciting to anybody sort of before this and hasn't been since. It is an interesting protein. I have been able to read about it quite a bit and learn more about it. But in essence, Dr. Wang starts to sort of think about this protein broadly. And he doesn't start by thinking about it as an Alzheimer's drug, which is okay. We shouldn't accuse anybody of doing anything wrong just because they changed their mind or their new evidence came to light. But just keep in mind that when he starts looking at this protein, he's looking at it because he thinks naloxone or naltrexone, I think it's naltrexone, binds to this protein. And specifically that it binds to this, what he calls a pentapeptide region, which is not the way I would call it, or any other structural biologist would call it, but this 5 amino acid region that's sort of faces water, so it's solvent facing. It doesn't face inward, and it is not clear what that region of that protein does at all. But he suspects that this is where naloxone, naltrexone, maybe other drugs bind to filament A. And usually determining that's pretty important in the world of chemistry and medicine. And we usually do that with some kind of structural biology. And what does that mean? So proteins are just amino acid sequences, right? So they're just strings of amino acids. And after around 40 amino acids, before then we call those peptides. After around 40, these peptides start to fold and make complex shapes. And we call those proteins. And so whenever you eat protein or something like that, this is kind of what we're talking about. And proteins are the entities that work, they do the work in the human body. We have all kinds of proteins. We have receptors, we have structural proteins, we have enzymes, we have just about 20,000 proteins, maybe more, that do all of the work of the body. They organize the DNA, they make your cells move, they harness energy, they store energy. Everything you can imagine is done by a protein in the human body. That's why they are the targets, almost always the target of a drug. Probably 80 or 90% of drugs target proteins. So There are some other exceptions, but that is generally the way it works. So he says this drug, or not this drug, but actually the well-known drug, naltrexone, targets the filament A protein. And that's actually how it works. Well, that's a big deal because it really seems like this opioid drug that has, opioid type actions, it reverses the action of drugs that target the opioids. It really would seem like that it would work through the opioids. Why would it work through a receptor that wasn't the opioid receptor? It doesn't make any sense. And he says, well, it's all because of this filament A. And none of this makes any sense. What would have been a cool experiment if he said, I'm going to shut down, somehow the opioid receptors and then show that I'm still getting some kind of effect just with filament A. And he hadn't really been able to do that, I don't think, because it would have been a breakthrough. And in fact, I think what he was trying to do was create a pain drug, an analgesic, that would work without touching the opioid receptor. And instead it would work through touching the filament receptor. Well, that would have been great. The whole world, you need to be living under a rock not to know that we're all looking for a great pain reliever that's not an opioid touching medicine. And there's a company called Vertex, which I've been following basically since I started following biotechs in the 90s as a teenager. But Vertex seems to have finally cracked the code with sodium channel inhibitors that are an amazing body of work where many pharmas have tried to drug the sodium channel and have failed. And finally, Vertex is about to get FDA approval for the first non-opioid pain reliever, which has opioid strength, but maybe not the liabilities of the opioids. So this is a big prize. I mean, that could be one of the biggest drugs ever. That could be as big as Ozempic, I think. But little old Dr. Wang, who really didn't have a career of note before this, decides that this is how these drugs work, and I can make a pain reliever from this. So presumably he's doing this for pain therapeutics, right? Your company's called Pain Therapeutics. You want an analgesic that's not an opioid. You want to solve the opioid crisis. They were first doing that by making a drug that could not be abused. That was called 𝗪𝗏𝗄𝐻. They seem to piss the FDA off pretty bad with 𝗪𝗏𝗄𝐻. Some people who were long Cassava or Pain Therapeutics think that there's a conspiracy there, some kind of Purdue level conspiracy to stop 𝗪𝗏𝗄𝐻 from working. By the time 𝗪𝗏𝗄𝐻 was filed, Purdue had lost a lot of power, or it was never really a big drug company to begin with, but it certainly lost quite a bit of power by then. And it was basically hurtling towards bankruptcy when 𝗪𝗏𝗄𝐻 was getting rejected by FDA. And the opioid crisis was in full swing, and there was a desperation for anything that could help. The problem seems to be that the company could not satisfy the FDA's analytical chemistry requests and solubility requests, which were like fairly basic, mundane things. And if you look at the history of the people that have worked at Pain Therapeutics, what you'll find is that they're generally not very experienced or qualified. And I think that their inability to address the FDA was mostly just kind of hiring the wrong people. I'm not sure 𝗪𝗏𝗄𝐻 would have been that great of a drug in the first place, but regardless, it was just kind of not all there. The same thing applied to their other two drugs. I was mentioning Gerson-Lerman earlier for a reason. So Pain Therapeutics back then had this ultra low dose naltrexone. And I was sort of laughing at the time because I said, well, why is it ultra low dose? Why shouldn't it just be naltrexone or high dose naltrexone? Does it make a difference? And there was this peculiar sort of answer was from Pain Therapeutics was like, yes, it actually has a completely different effect at an extremely low dose than a high dose. And I was 23 at the time or something like that. And I said, well, that's really weird, but maybe I'll accept that. And today at 41, I can tell you that it's complete hogwash. There's no drug that I'm aware of that has the opposite effect at some very low dose than and some other effect at some very high dose. It's just typically not the case. Could it possibly be true in 1 out of like 2,000 drugs? Maybe, but I've never really seen that. So anyway, I used Gerson-Lerman to try to figure out if their irritable bowel syndrome phase 3 would work. And they were doing ultra low dose naltrexone for IBS, big market, certainly could use a new drug. And one of the doctors in the IBS study told me that he thought it was the best drug he'd ever seen. He'd never seen anybody with IBS cured so quickly. And I said, well, how do you know it's not placebo? And he said, well, I'm jotting the patients down. And most of them have had no response. And then like a good chunk of my patients, maybe like a third to a half, have had these massive responses. It has to be that the drug works. And I told my boss, and he said, oh, that's really interesting. And I warned him that, look, anecdotes are kind of not that useful in medicine, and just it's just one doctor who, is probably just seeing things and hearing things. Of course, the trial failed. There was no effect whatsoever. I even think placebo is better than drug in this case. And it's very easy to, even be a teaching hospital clinician and not really understand that, the double blind makes it very hard to draw conclusions. So back to Dr. Wang. I just want to close the loop on that story. So back to Dr. Wang, there's this effort to maybe make a super pain reliever, and that was more or less abandoned. I do think at some point some chemistry work was done. There's a guy named Andrei Blasko, which I think very few people have talked to or thought about in this story. I have some chemistry patents myself. I love chemistry. Pharmaceuticals is literally the science of chemistry. So for me, it made sense for me somewhere around 20 years ago, even like to high school and college, but somewhere around 20 years ago, it sort of made sense to me to say, learn as much as you can about chemistry. Now, that didn't make me a full-blown chemist, but I'm able to enjoy a lot of great friendships with medicinal chemists, many great chemists I've worked with, including one of the Vertex chemists who worked on their cystic fibrosis medicines, who I would end up hiring to help lead my medicinal chemistry team. So I also have made chemistry discovery software. I feel like I'm decent at this space. And so at some point, I wanted to understand who actually made Cimiflam. Because if you look at the background of the people at Pain Therapeutics, there is no medicinal chemist to speak of. There's some process chemists, but that's not medicinal chemistry. So who actually designed and discovered this drug? And Blasko's name comes up. Now, Blasko's an analytic chemistry guy. He makes HPLC methods, he does COVAs, that kind of stuff. That's what he did at Novartis when he left Pain Therapeutics. But his name is on the patent. There's another name on the patent who I've yet to check out. But I tried to make contact with Dr. Blasko. He's long since gone from Pain Therapeutics. And I wanted to get a sense for how did that work sort of come about. Dr. Wang's name is on the patent. Dr. Wang, of course, not a chemist at all. Never wrote a paper on chemistry. Dr. Burns, same thing. In fact, all of Dr. Burns and Dr. Wang's papers have been retracted, which is the ultimate scientific embarrassment. It kind of takes a lot to have your papers retracted, and it's rather embarrassing, and all of their papers have been retracted, so they're kind of like these unpublished scientists at this point. And so where's the chemistry? Blasko, not really a medicinal chemist. So we're left with one last guy who's I'm trying to track down. But it really does matter because, again, drugs just don't sort of come out of nowhere and start working. They have to be exquisitely designed. And when we talk about what kind of drug this is supposed to be, this is really going to jump out at you. So there's this 5-peptide region, or 5-amino acid region. It's not a peptide at all. It's part of a large functional protein of filament A. Filament A is a monster protein. It is probably, if I had to guess, in the top 1% of all proteins by size. So it seems to do a lot of things, but it also seems somewhat redundant. But it does do quite a few things. In general, it seems to bind to actin, which is part of the cytoskeleton. Cells would be these like very shapeless blobs without things like actin, dynamin, all the other proteins like dystrophin, titin. There's a huge number of structural proteins that give the cell shape. And when those proteins are malformed or mutated, they generally don't work, and they cause a lot of catastrophic problems, especially in like muscle cells and things like that. So in any event, Dr. Wang says, this is the place. This is the place if you touch or stop or whatever, I'm not really sure what happens here, you block this site of this protein, you have the best pain reliever known to man. Doesn't make too much sense given this is a cytoskeletal protein, and doesn't sort of stand to reason too much. And that drug never makes it into humans. And Pain Therapeutics just sort of doesn't do anything with it. Wang files other patents stating that if you hit this protein in that spot, you stop cancer. And he also says that you stop diabetes. And he also says that you stop Alzheimer's. There are proteins that are important out there that touch on everything, but it's pretty rare for one protein to do everything. Now, having said that, maybe this is the one exception, and we're all going to be really surprised. But as we go peel the story back, we think about why this 5-amino acid section of a protein when there's 2,700 amino acids anywhere could be the right place to hit. And we have no idea why these 5? And I don't think Dr. Wang ever thought about it either, because there is a part of the job of people in medicinal chemistry teams is to do structural biology. And generally speaking, the best tool we have is crystal structures. You can also do things like NMR, and there's also like cryo-electron microscopy, cryo-EM. But basically, you want to make sure if you're going to invest, I don't know, $200 or $300 million into a clinical trial, you want to make sure that you know that your drug is hitting that protein at the right space, at the right place. And if you don't, you can't tell that with certainty, why in the world would you invest hundreds of millions of dollars on a drug, right? It's kind of like, making that, I don't know, the rocket thing that Elon made, and not sort of thinking about some of these key features, like, is this where the rocket's going to go? Oh, I don't know. Let's just try it and see what happens. You would never do that. you would think about it really carefully and not embarrass Mr. Musk as an engineer. There, you would say, well, this is how it's going to go, and maybe I'll simulate it on a computer, and maybe we'll do a smaller scale of it. You do hundreds and hundreds and hundreds of experiments that were the standard experiments you would normally do in drug discovery. I created I'm sure a lot of you guys remember me for this fiasco regarding a drug called 𝗪𝗛𝗪, where I raised the price of 𝗪𝗪𝗪 by 50-fold. 𝗪𝗪𝗪 has a target itself. It's the drug is called pyrimethamine, and it binds to something called DHFR, which is dihydrofolate reductase. It's an enzyme. The active site of that enzyme is bound by pyrimethamine. The chemists that made it at the time didn't really know what they were looking for. The tools weren't available. So what they did is they made a drug that bound not only DHFR's active site for humans, but also for toxoplasma, which is the bug you're trying to kill in this disease. So unfortunately, even though it stopped the active site of toxoplasma, it also stops the active site of humans, and it's a really important protein for humans to live. So you have to take this drug, and it's fairly toxic, and it's out of your system, and the infection's gone, and you're happy. So what we did is we designed the first ever, and you can look this up, in 𝗤𝗏𝐻𝐶𝐻. I'm not an author on the paper because I think it's poor taste for the CEO of the company to put his name on a paper or a patent. Unless they're truly a bona fide inventor, which I have some patents where I was truly the bona fide inventor, but I think it's just wrong to steal the thunder from your colleagues by putting your name on there. You're the CEO, you have plenty of thunder and plenty of accolades. You don't need to crowd out their scientific achievement. So you can read our papers in 𝗤𝗏𝐻𝐶𝐻 and other illustrious papers. This really isn't a better journal than 𝗤𝗏𝐻𝐶 for medicinal chemistry. And we made a drug that bound T-Gandhi DHFR, so toxoplasma, this brain parasite, and it did not bind the human one. And this was difficult. We had to do crystal structures. We took the first ever crystal structure of human T-Gandhi bound to DHFR, if I'm not mistaken. And we took the first ever crystal structure of it bound to an inhibitor that doesn't bind the T-Gandhi. This is a basic drug discovery. This is not Nobel Prize winning work. We used a company called 𝗔𝗍. It cost about $50,000 to do the co-crystal. It was not $5 million or $50 million. We were a small private company. It wasn't that big of a deal. Anybody could have done this, but Pain Therapeutics didn't. And I think that, again, if you look at some towering accomplishment of engineering, and you say, well, this was designed without a blueprint, you would say, no way. You can't design a building like this without a blueprint. One person designed all of this without a blueprint. This beautiful skyscraper, impossible. It is 1,000 times harder to do this in medicine. And we tend to trivialize drugs as, oh yeah, this pill might work. It really is a marvel of engineering to make a medicine work. And you need to do these sophisticated, or relatively sophisticated, sort of mechanisms or methods, I should say, to actually make something happen. They don't want to be anybody that, is below what they think they're capable of. But unfortunately, sometimes we're not capable of things that we dream of, or we may be capable of them years from now, or something like that. But we want shortcuts. We want to be in charge now. You feel you're good enough to be in charge. Why aren't you in charge? So when you send back a result and it's, oh, this compound didn't work. Well, maybe Pain Therapeutics says, we can find another lab to do this work. And you're at CUNY, you're not at Harvard. There's a chance your lab's going to get closed if you don't get NIH grants. There's a chance your lab's going to get closed if you don't get corporate grants. So you say, you know what, guys? This drug worked amazing. Isn't that great? Look at that. and you don't really care if this drug goes into humans because your job is to impress your wife or impress somebody else. I don't know what. Only Wang couldn't talk about it, and he'll probably have to talk about it when he pleads guilty to fraud, or when he is found guilty of fraud and is sentenced. Wang's conviction is really not in doubt. Unfortunately, I know from personal experience that the likelihood of being convicted in criminal court is close to 100%, in federal criminal court at least. It's basically impossible to win. You can really go back in time and find very, very few cases where you actually win in federal court. Trust me, I looked myself, and I tried to hire the attorneys that actually deliver those wins. And Dr. Wang is not just unlikely by the odds to lose, but a group of fellows who I have no affiliation with or knowledge of or friendship with or any constant contact with. I didn't know about their campaign. But they made a site called CassavaFraud.com, and you actually just can see for your own eyes what Dr. Wang did. He took the images for the Western blot, which is this basic science experiment that's very important. It's kind of like a screwdriver for scientists. And he took the Western blot and he just did basic image manipulation with a tool like Photoshop. I mean, you can see it with your own eyes, and it's insane. When you look at it and you say, you were trying to make the like I said, something that's more difficult than making the rocket thing that Elon made, the rocket self-landing. You tried to do something like that, but you faked all of it. Instead of a CAD CAM or whatever tools that you would use for engineering, you just basically winged it, and you did it for whatever reason, ego, who knows, and you just faked it. And to me, that just ends the story right there. But it actually gets worse, which is the craziest part. I have more research to do on the preclinical work. I've tried to understand filament A better. I've tried to look at Wang's work. It's kind of a little inscrutable. The most amount of work that Wang did that was like published quality and like reviewable is in the patents. And reading those is a little difficult as well. It's sometimes not really clear what he's trying to accomplish in his preclinical work. It's certainly not the work of a drug discovery guy. Drug discovery people do different work. In some ways, he's mostly trying to prove filament A and this other protein called alpha-7 react or interact. And that's not really kind of the point of most of this work. So the actual cemiphilam work is just not really there or clear as to what exactly the result he's accomplishing. And again, this really isn't complicated stuff. I mean, usually what you're trying to do is you're trying to stop some biological signal from happening. And the amount of drug it takes to do that is what's sometimes called potency. So if it takes 1 nanomole of drug to stop a receptor from activating or an enzymatic reaction from happening, then you sometimes say your drug is 1 nanomolar potent. A nanomole is a very small amount. If your molecular weight is 16, for example, I think you're looking at a very small very small amount of drug, for example, 16 nanograms, to actually block that interaction. And that's something that you could deliver in, say, a 50 milligram pill. And this is the science of pharmaceutics. It's just sort of well known, and a lot of those things were not done by Wang. And I think it's because the guy never made a drug in his life. He's not a chemist. He's not even a pharmaceutical guy. So where were the scientists at Pain? And I think one of the problems with Pain Therapeutics is they never had a chief scientific officer. They had Remy, who is not a scientist at all, and not really a self-taught scientist. Again, I know Remy. There's nothing about Remy that screams scientist to me. So Remy's not a self-taught chemist or self-taught chief scientific officer. But even if he was, look, I think I've learned a lot about this field. I had a great chief scientific officer named Dr. Andrew Vino, who is a medicinal chemist that worked under a very, very famous chemist who won 2 Nobel Prizes, Barry Sharpless. Barry is one of the most important and famous chemists in the world. Andrew was his postdoc student. Andrew worked at some drug companies. He founded his own drug company after working with me. That's doing its IPO. He's done very well. But I also had somebody that worked above Andrew named Dr. Maria Bocconi, who is not a chemist, but she was my chief scientific officer, who was more of a preclinical biochemist. And she worked at Merck, and she was responsible for a lot of big work there. You know, you got to have somebody that you can point to that knows science, even if it's yourself. In my case, I think people tell you I know science pretty well. You still want somebody that this is their thing. They didn't learn it on their own. They did this for a living for a long time. And, you know, even I had that. And Remy has, at most, a chief medical officer. Now, I had one of those too. You need somebody to run clinical trials. But the people that run clinical trials don't know science. They know medicine. Those are different things. So in any event, you know, the company was just sort of this conspicuous absence of actual people who knew how to discover and develop drugs. But Wang sort of gave them 𝗪𝗡, and the company, after failing all their pain trials, they said, we're going to change our name to Cassava, and we're going to try to see if 𝗪𝗡 is the cure for Alzheimer's. And I'm being a little tongue-in-cheek here because it's without doing really advanced scientific work. Like if you look at a company like Denali, this is a company that spun out of Roche, hundreds of millions of capital raised, real science work going on there, still a long shot, but, you know, maybe, who knows, they can come up with a treatment for Alzheimer's. But to take a company that actually doesn't have any actual scientific capability, that would seem very difficult. And to outsource that scientific capability to a guy at CUNY who's not a chemist, who would end up having his reputation tarnished, including having the really uncommon situation, I think maybe one of the first ever, being arrested. I think there's 2 or 3 others, but being arrested for faking your scientific data. And again, still no chemist in sight. I think it's just very odd. So anyway, they say, let's throw caution to the wind and let's put 𝗪𝗡 in a trial. Now, to go into a clinical trial, you have to do something called toxicology. And when you do toxicology, you do generally a battery of other experiments. And one of the things you do is you try to figure out the disposition of the drug. So how do you do that? Well, you do this in toxicology, and you do this in other ways. But basically, you take a bunch of animals and you euthanize them. It's pretty morbid. And you try to figure out how much drug exists in the homogenate of those organs. So you can do this through LC-MS, for example, and you create an analytic method. And you validate the method, and you check to see how much drug ended up in the liver, the spleen, the brain, the lungs, the skin, the eye. There's like 36 organs that you try. And there's actually people who do this for a living, and they have a degree called a DVM, which is a doctorate of veterinary medicine. And the fellow I mentioned earlier from Merck, who started his own consultancy after working at Merck for a long time, who was thanked in the book I mentioned, he was my guy that helped me do this. And he was shocked at how much I knew about this. And he said, how do you know so much about this? And I said, Jim. His name's Jim McDonald, Dr. Jim McDonald, who I'd highly recommend, who, you know, did all the work for a lot of the Merck legendary drugs like Zocor, for example, which is one of the biggest drugs ever made, and many others. And I said, look, I'd like to learn this stuff. If you can do it, why can't I do it? And he said, it's a great attitude. And I tried to learn as much as I could. I'm not Jim, but I know, you know, a fraction of what he knows, and that's better than knowing nothing. And so anyway, the DVMs and their counterparts other scientists, they try to, with analytical chemistry, try to figure out, where did this drug go? It went into the animal. It came out somewhere. It either comes out in feces or it comes out in urine, and you test that very carefully. But some of it stays in the animal at the time of euthanization. And knowing where it traffics to and how is an art, you know, there's a number of ways to do it, including with radio label drug. And what was interesting, which I didn't know, is that in the investigator's brochure that Cassava supplied, and this is like, I don't know if it's breaking news or not, but I don't think this is well known. But in Cassava, whenever you do a clinical trial, you supply something called an investigator's brochure to the doctor who's willing to do the clinical trial. Doctors don't do clinical trials in their ordinary work. It's unusual. They see patients. That's their job. To do a clinical trial is really painful. You got to do a lot of paperwork. You got to fill out a lot of stuff. You got to track the patients really carefully. You'd rather be treating patients and just seeing patients and doing your normal, you know, appointments. They make more money that way. So you got to pay doctors a bunch of money, and they got to have the staff, and they got to have the specialty. But some doctors like to do it, and they do it. So in any event, to do that, you give out this investigator brochure. It has some details about the drug, how is it discovered, what's it supposed to do, blah, blah, blah. And in the investigator's brochure that, a contact sent me, it says that in their animal experiments, that the brain was one of the organs with the least amount of drug exposure, and that all the traditional suspects like the lung and the liver and all the other places you would normally expect a drug to show up, like the spleen or the intestines, these had all the semiphlam, but the brain didn't have any. I'm perplexed by this because typically for a brain disease like Alzheimer's, you would need your drug to go to the brain. I would say that's, a requirement. And sometimes drugs can get into the brain, but the brain is a very hard place for drugs to go to. Why? There's these things called endothelial tight junctions, which some people call the blood-brain barrier. And this stops just any old molecule from entering the brain. Sometimes you need active transport through a receptor. Sometimes you can get passive transport if you have a small enough molecular weight and you have, these are all, by the way, necessary conditions, and you're not like, you have to be lipophilic. You can't be too hydrophilic or polar. You have to be relatively nonpolar. And most importantly, you have to not be a PGP substrate or an extrusion protein substrate. So there's a bunch of channels there. They'll actually kick you back out. They're like bouncers for the brain. They'll kick your drug back out. And there's a whole bunch of those. So it's not a guarantee that you can get into the brain by any means. And you have to, this is why we do preclinical chemistry. This is why we do preclinical drug discovery. All of these things you would normally do, it's not really clear that they did, but they did do this. And we know that the drug doesn't end up in the brain too much, at least, which is very, very, very weird. And that's in black and white in Cassava's own writing. So how much exactly gets into the brain or not, we don't know, but it doesn't seem too great. The other thing you do in drug discovery, and we're just dancing around right before the human experience, which is the second half of the story, which is very interesting. So we're ending the kind of the first half. But the other thing you do in chemistry that's not very hard is you make sure you have good pharmaceutical properties. What are good pharmaceutical properties? Well, good bioavailability. We don't know what the bioavailability of this compound is. It's never been disclosed. You have a good half-life. We know the half-life of this product is 4 hours, which is pretty much as low as it gets. Like most modern medicines for small molecules, 4-hour half-life's pretty close to the borderline of useless. You have to build up what's called a steady state concentration, sometimes called Cmin. There's Cmax, pardon the crude jokes, but Cmin is capital C, lowercase min, and it's the minimum concentration in the blood for a drug. It's actually quite important, especially for this proposed mechanism. If you have any period of time where you do not have drug on board, that there is some period of time where there is no drug in your system because the Cmin is close to 0. Cmax peaks. Whenever you take a drug, whether it's your antidepressant or your antacid or whatever, the Cmax is like 1 hour or 2 hours. As soon as you take that pill, bam, there's a huge leap in concentration and it drops just as quickly, but there's some that stays there, and that's the idea of the half-life. And you want that half-life to be as long as possible in most cases, because you don't have to take the pill over and over and over and over again. Nevertheless, even with 8-hour, 10-hour, 12-hour half-lives, we still have to do controlled release. We still have to do other mechanisms, because it's not just a half-life. There's other properties of like volume of distribution and other things that are important to think about for what kind of dosing do we want. So it's not clear that, for example, let's say, I don't know, you had a cancer receptor and you were designing a drug, and as long as you block that receptor, you would stop the cancer. Well, you would want to make sure that receptor was blocked 24/7, literally all the time at maximum amount. Because let's say if some binding agent would naturally bind that and cause the cancer, you want to make sure that would never happen. Even if the patient maybe missed a couple doses, you want to make sure that doesn't happen. Because then chaos would ensue. And it's really not clear why they have this dose and why, they thought that they're actually getting much drug into the brain, or why this half-life, which is so low, could even be useful. So you can do some of these basic calculations with a basic textbook on pharmacology, and I think you'll be surprised.